This is a stem cell. Essentially, it serves as an internal repair system without limits to what it can become. Stem cells divide through mitosis or cell division. Stem cells are totipotent. They can develop into a variety of cells, including blood, brain, and muscle cells. This process renews and repairs tissues and organs and can quickly help regenerate different cells through proliferation. Proliferation is the rapid reproduction of the cells, an extension to mitosis. There are two main groups of stem cells, embryonic cells and somatic cells. Embryonic stem cells emerge from cells from an embryo. Embryonic stem cells are pluripotent, capable of rising to different cell types. Embryonic cells were first discovered in mouse embryos in 1981 and have since been acquired from human embryos starting in 1991. Despite the stem cell's potential to solve a multitude of human health problems, obtaining embryonic stem cells can be a challenge when analyzing the ethics of obtaining human embryos, considering that embryonic cells are often taken from aborted fetuses. The other group of stem cells is composed of somatic stem cells. Unlike embryonic stem cells, the origin of somatic stem cells in some mature tissues is still under investigation. Somatic stem cells are undifferentiated cells which can specialize into different cell types, but are more limited compared to embryonic cells. Several questions have arisen in current investigations. Firstly, it is unknown why embryonic stem cells can proliferate for a year or more in the laboratory without differentiating, but most adult stem cells cannot. Secondly, it is unclear what the factors are in living organisms that normally regulate stem cell proliferation and self-renewal. So why are we interested in stem cells? Primarily, stem cells help us understand how diseases occur. By observing stem cells develop new body parts, researchers and doctors may gain a better understanding of how diseases occur. Also, stem cells can be used to test new drugs for safety. Human stem cells can be programmed into tissue-specific cells that can be used as test subjects. For example, stem cells can be used to make a nerve tissue to test a drug for a nerve disease. And finally, stem cells are able to generate healthy cells to replace disabled ones. This opens up a new method of medical treatment that can be used to cure more substantial diseases. We have only scratched the surface in observing the vast potential of stem cells. Further development indicates the beginning of stem cell treatment being widely used to effectively cure various diseases.